and I'm sure it is. May Allah help everybody. Bad. I would like to say that I hope you took part in yesterday's demonstrations, inshallah, Quds Day. I hope that is also Kabul. We have lost a very great filmmaker in Iran called Mr. Nader Talebzadeh, who was a very, inshallah, who was a very active anti-Zionist element. May Allah bless his pure soul. Please recite Fatiha for him. His name is Nader Talebzadeh. He was a very great man, and he died on Al-Quds Day. He was very much anti-Zionist. Anyway, I hope it's Siyam Maqul. Thank you very much to all of you. I'll just be with you in a minute. Okay, see, from the last week's lecture, there were 2 to 3 points which were not very clear. So I just thought I will just go over the lecture once more with you about the kinetocoprotein formation and damp proteins and YACs, yeast artificial chromosome. I just thought I'd just review it once more with you, and then we'll go on to the new ones, to the new slides. So we just said that there's a severe problem of packaging, histone marks, active and inactive. Histone codes mark active and inactive sequences by acetylation, methylation. DNA elements for chromostructure kinetics include ARS, which was autonomous replication sites, telomeres and centromeres. Very important. CEN or centromeres promote the assembly of the kinetochore, which is a giant protein complex that attaches the chromosome to the spindle at division. And we said the largest chromosome is 3 × 10⁸ base pairs. Severe problem of packaging is like, as we said, by typical cell is 10 micrometers, and then here it is like packaging 6 foot long string, mile, 11-mile long string into a 6 foot box. Visualizing chromatin breakdowns, beads on a string with low salt, high salt. We said that they contained H1, H2, H3, H2A, H2B, H3, and H4. Then we said here that they discovered this by first they took the packaging protein was discovered by DNA, the one digestion of the nuclei, and then they saw that after they digested it and they ran it on agarose gel, they saw multiples of 150 to 200 base pairs. So they said something must be protecting the DNA. Then they realized that yes, the protecting the DNA were the protein, the histones. Packaging protein discovered by DNA is one, digests of nuclei, nuclei isolated, non-specific endonucleases. Once more, when they were treated with more DNAs, multiples of 10 base pairs came out, and they realized that the histones had gone. Crystal structure of the nucleosome, ribbon drawings, top views, 147 base pairs of DNA wrapped almost twice around 8 core host histones, H2A, H2B, H3, and H4. The histone fold H3 and H4 tetramer binds to H2 or H2B to form the histone opsomer. This we know very well. Because the H3 and H4 are there, then the DNA is not smoothly bent because we said because of hydrogen bondings. Water mediates many histone phosphate contacts. Water on the DNA. Then we said histone tails stick out between the guides of DNA and they attach themselves to the next, and then they get with the histone code when they're acetylated, when they're methylated, gene silencing, heterochromatin, phosphorylated, mitosis, meiosis, expression takes place, histone depletion, all these, we said this is on the lysine and the serine. Now, hypoacylated histones and heterochromatin, when they had CIR2 proteins, CIR proteins bind cooperatively to deacetylated chromatin. CIR2 is a histone deacetylase. The combined action of the packaging proteins and the deacetylase spread heterochromatin and silence package genes. So whenever CIR2 protein is there, you get heterochromatin. They silence the package genes. Then we said that there was an experiment where they took the replicates of mixed chromatin and nucleosome-free templates in vitro, and after the replication, they saw that only the ones which contained mixed chromatin, this was a only the mixed chromatin showed nucleosomes were found only on the daughter of the mixed chromatins and not on DNA templates. So it is realized it was something other than DNA. Then we said larger DNA structures mediate more compaction, euchromatin, transcribed and less condensed loops of this. All this we have studied before. Heterochromatin, more condensed, gene silence, replicated, and S phase. DNA amplification produces visible balance in staining pattern by fly polythene chromosome. You remember the polythene chromosome we discussed? We said it was Drosophila. Good morning. Then important now here, we come to the important matters. Telomeres, centromere. We said that the functional DNA sites are telomere and centromeres. Schematic and electron micrograph of X chromosomes. Telomeres protect the ends, and centromere is at the primary constriction. It mediates chromosome cohesion, spindle attachment, and chromosome segregation. Now we will go into more detail. Kinetochores mediate chromosome microtubule attachment. See? Centromere DNA segment is at the primary constriction. The kinetochore is a huge, complicated protein complex with several layers. The outer layer provides attachment sites for microtubules. Now, centromeres contain special DNA sequences that assemble centromeres. Now here, what we're going to discuss. Model for yeast kinetochore, microtubules, and the DNA. You remember we did this from the last cycle. We said in the M, G1, S, and G2. In G1, the cell prepares for cell division. In S, DNA replication takes place. In G2, chromosome segregation takes place. To prepare for chromosome segregation. And in M, chromosome segregation, mitosis, telokinasis, and it breaks up. So you have G, S, G1, S, G2, M. Now, the events of mitosis. You have the kinetochore, cohesion, microtubules, then they pull them apart. The cohesion proteins break up. And we said that if there's more than one centromere, chromosomes will break up. If there's no centromere, it is not, they do not divide properly, or they're not given properly. Okay. Now we said here, now this is what we're going to discuss. Histone variants alter nucleosome function. H2AZ histone inhibit nucleosome from forming repressive chromatin structures. They create regions of easily accessible chromatin that are more compatible with transcription. Now, they prevent heterochromatin being formed. Now here, CENPA. CENPA replaces H3 and is associated with nucleosomes that include centromeric DNA. Interact, this one, H3 comes out and interacts with the kinetochore protein, okay? Now there was an article in the Nucleus magazine journal. Good morning. Where we say H33 is deposited at centromeres in S phase as a placeholder for newly assembled CENP in G1 phase. Now this is very interesting. We'll go on and you'll see why. Give me just a second. Sorry. Now, I'm sorry. H33 is deposited at centromere in S phase as a placeholder for newly assembled CNPA in the G1 phase. You remember the phases which were discussed here? G1 phase, S phase, G2, M phase. So the centromeres here in the late M phase. but they are synthesized earlier. Special DNA sequences ensure proper chromosome sequencing. Centromere identity is defined epigenetically by presence of centromeric histone H3 variant CENH3 called CENPA in humans. H3 variant is replicated in the S phase, but the newly synthesized CNAPA is deposited in the late telophase. Now these observations, okay, fine. Now we are talking about, I know that. What we are talking about is the histones, because it's a special histone called CENPA, which is deposited later. But as replication proceeds, the histones have to be available for the DNA to wind around it. So this is what happens. Understanding such molecules, what happens is ordinary H3 nucleosomes are put in, and then the H3 nucleosome gets removed and the CENPA will come later on. This is what it's trying to say. Understanding such molecular mechanisms is important to reveal the composition organization of centromeres in mitosis when the kinetochore forms and functions. Here we investigate centromeric chromatin status during the cell cycle using the snap tag methodology to visualize old and new histones on extended chromatin fibers in human cells. Our results show that both histone H3 variant, H31 and H33, are deposited at centromeric domain in S phase. There is reduced H33 but not reduced H31 at centromeres in G1 phase compared to S phase. These observations are consistent with the replacement model, where both H31 and H33 are deposited at centromeres in S phase, and placeholder H33 is replaced with CENPA in G1 phase. So what happens is, it's trying to say that when replication is taking place, even though the CENPA, even though the CENPA is formed, it is not deposited. Because it is not deposited, it's deposited much later because it has to attach with the kinetochore proteins. So meanwhile, the variant of H3A and H3B, H33A, is it? What is the variant? H31 and H33 are deposited. And later on, the H3, H31 move out and they're replaced with the CENPA. This was a big experiment which they did to find this. And here is your CNPA bonds interspersed with H31 and H33, DNA replication, nucleosome aggregation, S phase, CNPA, because CNPA gets diluted. The new ones are not deposited, so it gets diluted. So meanwhile, H3 will come and new H1 and H3 is deposited, then H31 is removed, and CA goes in. So what happens? Model for dynamics of CNPA. H31 and H33 deposition at centromeres during the cell cycle. CNPA blocks at centromeres are interspersed. Okay, we are talking about we are discussing the dynamics during replication. The protein comes on, it is synthesized early. Okay, fine. I didn't ask you that question. I just want you to understand this, that they get, while during transcription, what happens is the CE and PA blocks are interspersed with H31 blocks. See? See, and H31 blocks, old H3, new H3, H31, H33. Now, because when it is replicating, the new CNPA is not replaced. So because it is diluted, they get replaced by H31 and H33. Then when they enter into the G2 phase and mitosis, they are removed and instead the CENPA will come. That is all it's trying to say. Very important it is, okay? CNPA nucleosomes are redistributed to daughter strands and are diluted by half. Newly synthesized H31 and H33 are deposited as placeholders at CNPA domains and interspersed domains. Half of new H31 is deposited at CNPA domains and half at interspace domains, whereas new H33 is most frequently deposited at interspace domains than at CNPA domains. After S phase, half of H31 at centromeres is new, whereas most H33 at centromeres is newly deposited. During G2 phase or mitosis, H33 may be removed, is replaced by new CNPA. The amount of H33 at centromeres during G1 is reduced compared to S phase, whereas H31 does not change. New CNPA may not always occur at the same centromeric position during every cell cycle. So why are you making so much of noise? We know all this. We know it's a centromere protein. We know it attaches to the kinetochore. But we were discussing that during replication, even though they are synthesized early, they get deposited in the histones, in the nucleosome late. So meanwhile, H33 and H31 replace them. When they get replaced, after some time, the H31, H33 is removed and CNPA comes in its place. And in every cell cycle, it doesn't happen in the same position. That's all. It's clear? So much of noise. Is it clear? Okay. Now, here kinetochore complexity and structural challenge. See the kinetochore? Even in the yeast, there's a thing called the dam complex where all these other complexes will come and sit next to it. Dam complex has got 10, it's a decamer, 10 subunits. Now, what does the dam complex do? Like it forms rings around the kinetochore and helps in pulling it away from each other. Components of the dash complex, a microtubule binding subcomplex of the outer kinetochore. In the yeast is called dam complex. In eukaryote, in others it's called dash. Essential for proper chromosome segregation. The dash complex mediates the formation and maintenance of bipolar kinetochore microtubular attachments by forming closed rings around spindle microtubes and establishing interactions with proteins from the central kinetochore. Okay. The dash ring complex may both stabilize microtubules during chromosome attachment in anaphase A and allow the chromosome to remain attached to the depolymerizing microtubule in anaphase B. Microtubule depolymerization proceeds by protofilament splaying and induces the kinetochore attached ring to slide longitudinally, therefore by helping to transduce depolymerizing energy into pulling forces to disjoin the chromatids. So what happens is they form this ring and they slide along. They slide along. I'll read it once more. Dash complex mediates the formation and maintenance of bipolar kinetochore microtubule attachments by forming closed rings around spindle microtubules and establishing interactions with proteins from the central kinetochore. Okay? So they form microtubule maintenance of a closed ring around the spindle microtubules and establish interactions with proteins. The dash ring complex may both stabilize microtubules during chromosome attachment in anaphase A and allow the chromosome to remain attached to the depolymerizing microtubule in anaphase B. Means that the microtubule is getting smaller and smaller. It's getting depolymerized in anaphase B. Microtubule depolymerization proceeds by protofilament splaying and induces the kinetochore attached ring to slide longitudinally, thereby helping to transduce depolymerization energy into pulling forces to disjoin. So what it does, it helps it to slide longitudinally and helps to get the energy into pulling to disjoin the chromatids. So what happens is the two sister chromatids are joined together. Let's see, there was this picture here, I think, if I'm not mistaken. Yeah. They join together, and then you have this dash complex which comes here like rings. The dash complex forms it attaches to the proteins on the kinetochore, and it helps, it slides along. It slides along as the microtubule gets depolymerized. It gets smaller and smaller. As it gets smaller and smaller, the chromatids are pulled far away from each other. Okay? Got it? Okay, because I felt the last time we were not very clear about this. No. There's also a thing called here it is, the pictures here, see? Dam rings slide along shrinking microtubules. There was also another thing called the yeast artificial chromosome, which we said comprises of, what this does is that it can, a lot of human chromosomes, DNA can be up to 1 million nucleotide pairs can come here, and then they were studied for the Human Genome Project. Now, let's discuss a little bit of that. Yeast artificial chromosome is a human engineered DNA molecule used to clone DNA sequences in yeast cells. YACs are, I call it YAC. YACs are often used in connection with mapping and sequencing of genomes. Segments of an organism's DNA up to 1 million base pairs in length can be inserted into YACs. The short arm 5 kb contains 4 DNA elements derived from the YAC vector sequence, a functional telomere, a centromere, origin of replication, and a yeast selectable marker. Telomeres, a origin of centromeres, and yeast selectable markers. This was initially used for Human Genome Project, but due to problems with stability, a bacterial artificial chromosome is used. Primary components are S, centromeres and telomeres from S cerevisiae and selectable marker sequence, such as antibiotic resistance, and visible markers to select transformed yeast cells. So you require two things also. You require antibiotic resistance and a visible marker so that you can make out the telomeres will not be stable during extracellular repression and will not be distinguishing from other colonies. So they're used in connection with mapping and sequencing of genomes. Segments of an organism's DNA up to 1 million base pairs in length can be inserted into YAC. The short arm 5 kb contains 4 DNA elements derived from YAC vector sequence, a functional telomere, a centromere, origin of replication, and a selectable marker. This was used for Human Genome Project, but due to stability, it's down by bacterial artificial chromosome. Primary components are ARS, centromeres, and telomeres from S series C, and selectable marker sequence such as antibiotic resistance and a visible marker. So, problem is packaging. Yeah, but as it is, they're not very stable, they're not used much more. Problem is packaging, levels of chromatin structure, the telomeres, you mean, yeah? Histone codes active and inactive sequences, DNA elements. For chromosome structure includes ARS, telomeres, and centromeres. CEN promotes the assembly of the kinetochore, a giant protein complex that attaches the chromosome to the spindle at division. YACs contain centromeres, telomeres, ARS. Okay, that's all. Now, okay. I will send the file which is now modified to you once more. Now we will discuss chromatin assembly. Very important it is. See, as we are going on and on, we started off with amino acids, then we went on to and proteins, then we went on to transcription, then we went on to chromosomes, histone code, chromatins, telomeres, now kinetochore proteins. Now, I was going to discuss transcription factors with you today, but I thought it would be, considering it's the end of Ramadan and we're all fasting, so we'll do it for the next class, inshallah. So here today we will discuss, it's a little bit easier and interesting also, not at all. Chromatin assembly. Very important. We're going to discuss chromatin assembly during replication, nucleosome positioning, epigenetic inheritance, chromatin assembly during transcription. You know this by now quite well. Okay, Hinda, you're right. That's why they say aging is with telomeres, when the telomere of the chromosomes become a problem. Okay, fine. Now we will talk about nucleosome assembly. Thank you very much for all the information you have given me on telomeres. Now we come to, we've taken out long-range fiber-fiber interaction, chromonemar fibers, linker histones, short-range internucleosomal interactions, beads on a string when they open out, and the nucleosome, acetylated DNA methylation, they get repressed. Okay. Now, please pay attention to the class now, okay? You're distracting me and distracting everybody else. When I ask you questions, you can answer me. Thank you. Now, visualizing this primary structure. Now, it's very easy for us now to discuss histones, histone tetramer, H2A, H2B, H3, H4, very easy. But for people that have worked for many, many years to come up with this data. So, why no sound? There should be sound. No sound? Try hands-free, please. You can hear me? Okay. So, as I said, they have worked for many, many years to get the results. We are going to discuss all these histone chaperones. Very good. So you have we are going to discuss each histone chaperone today. I'm very glad you know about it. So post-shell macronucleus generates mono, di, and trinucleosomes. How is chromatin assembly achieved? Here's an experiment. They've added histone, DNA, and salts. And then you got your aggregates. Here they've added histone, DNA, and negatively charged proteins. You get mononucleosomes and poor nucleosome array. Can you tell me why? Tell me what, tell me. Answer my question. Okay. Baba, answer my question. I'm not asking you for lectures. Okay. Histones is positively charged, DNA is negatively charged. So negatively charged proteins, what happens? Histones will go and stick with this instead of sticking with this. Okay, so there is a competition between negatively charged proteins and DNA for histones. Okay? That's why you don't get a good assembly here. Very beautiful work, actually. I like the experiments because it shows that histones are also positively charged. And because the DNA is negatively charged and the inverse is opposite, they added DNA negatively charged proteins. And they saw that the assembly is irregular, not good assembly. A similar work was done with Xenopus drosophila. I think Abdul Kazim would like to give the lecture, yeah? Should I put it on and you can give us the lecture? Very nice. Xenopus drosophila nuclear extract chromatin are regularly spaced. In vitro, tetra, tri, di, mono. Okay. What is in the extract? Histone chaperones and chromatin remodeling activities are present in the extract. I will give you a chance, Abdul Kazim, maybe a little later. Last 10 minutes of the class, you can give us a lecture, okay? So, histone chaperones. So just now, stay calm till I finish. Don't distract me then, please. Chromatin remodeling. You can give us a lecture on the summary of what I teach. Histone chaperones, chromatin remodeling activities. Now, an in-house assembly, as Abdul Kazim has said, is achieved by chaperone nucleosome, and these are the chaperone proteins. PHD is the chromodomain helicase binding protein. ATP is required. Then, if one is there and one is not there, all three are there, one is not there, they do not form good nucleosome array. But when ATP, NAP1, and CHD1, this is the chromodomain helicase binding protein, histone chaperone, and ATP is given, you get good array. Very good array. Please note everything is duplicate. Enhanced assembly, when all three are. If one of them is missing, you don't get good assembly. So, chromatin disassembly when? During replication, transcription, and repair. Now the question is, why must there be disassembly? Why must there be disassembly during replication, etc.? Why must there be chromatin disassembly? No one answering me? Why must there be disassembly? Yeah, Hussein. I just asked a simple question. Exactly. To facilitate the movement of the DNA replication machine. We don't want such a long story. A close link between histone has been determined. We're not talking about histones. We're just saying that why must there be a chromatin disassembly? To facilitate the movement of the DNA replication machinery. Very good. Because if the DNA is wrapped very heavily, then it is not exposed and no replication can take place. So it has to be disassembled during replication, during transcription, and during repair. And we didn't need such a long story for that. Yeah, exactly. Now, we will talk about chromatin disassembly during replication. I think I will give you a 5-minute break.